this is the latest in the series of workshops that we at CICAG have organized. These started off as a part of an open chemical sciences meeting in which we had 3 different strands, one on open data, one on open publishing, and then this strand on open source software. The workshops on open source software proved to be incredibly popular, and the YouTube channel where we put all the YouTube videos has been visited hundreds and thousands of times. So we felt it was really important to try and continue this. We started off with the workshops this first half of the year, which again have continued to be really popular. And David Coase is going to be giving us this latest workshop on docking using GNINA. But we also organized a continued stream of workshops. So the next month is on Data Warrior, but we've also organized one a month going right through to December. So details are on the CICAG website, but you'd be more than welcome to have a look at those and continue these open source software workshops. These are only possible because of the great work of all the open source scientists, but also I want to acknowledge Liverpool Chirochem, who are actually funding the licenses, etc. we need to do these video workshops. So many thanks to them. Our format is that we'll do an hour of workshop and then we'll take a break for 5 minutes or so people can stretch their legs and then continue with the workshop. If you want to ask questions, there's a Q&A button at the bottom of the screen. Please use that rather than the chat, and we will monitor the Q&A and transfer the questions to David. We're lucky that we have Drew McNutt here who will actually be monitoring that channel, and anything that he can answer directly, I'll probably just reply within the chat. With that, I'd like to hand over to David, and I'm really looking forward to what looks like it's going to be a really interesting presentation and workshop. Over to you, David.
Great, thank you, and thank you for inviting me. I'm very happy to be here and present our GNINA docking software. This is the first time I have presented this material, so if anything is confusing, it's me, not you, so don't hesitate to use the Q&A to ask questions. This has been, okay, so first I should say, we should, there's this link here, but let me put the link to the slides in the chat. Where'd the chat go? Actually, Drew, could you just put the link to the slides in the chat?
I'll do it for you.
Oh, here it is. There it is. I got it. There we go.
Yeah.
All right, so you can follow along it. So I'm expecting you to follow along the slides. And there is a Colab notebook here where I've just pulled out all the Python and the things that you can actually run. So you can, run the code as we go through it in the slides. These slides, if you're not familiar with this format, these are Jupyter notebook slides. So actually this whole notebook is also available in Git if you want to go through it. This has been several years of work developing this software from many people. I'd like to acknowledge all the authors on the paper here. Drew is here in chat. Actually, I noticed that Rocco is signed in too. Chris, maybe if you could add him as a panelist.
Okay, I'll see if I can do that.
Also be able to answer questions, especially with regards to flexible docking. So before we even get started, why do you want to dock? So this is going to be a little bit interactive. What is your main interest in docking? Let's see if this works. So this just make sure everyone has the slides up and is following along. Try to engage the brain. We won't wait for it. Inverse virtual screening. Yeah, just pick your, click on it in the slides. Like so, predict pose, I'll submit. All right, so good. This is great. We'll go there. It's a power of two. Okay, so pose prediction, virtual screening, a little bit less affinity prediction. I'm glad no one said I don't know. Maybe, although you would find out, I guess. So of course it can be used for all of these things. Although I'm glad that pose prediction is popular because that's probably, that's sort of the main motivation for docking. Because when we say docking, the task is to predict the most likely conformation and pose of the ligand in a given binding site, right? So we have some structure and the crystal structures in these light sticks, and we're trying to sample all the conformational space and find that conformation and pose that scores the best, right? So this is the task. And so this scoring function is really important. At a minimum, it needs to score the right pose the best. Ideally, in the perfect world, the score would equal the binding affinity. And if we can't get that good, at least productively rank compounds for virtual screening. So like maybe we can't actually predict this as nanomolar, but we can at least identify the best affinity compounds. One philosophy I'd like to share with you is that this is a score, not an energy. I always get a little nervous when people start talking about docking energies and they lose the fact that it's a predicted energy, and then experimentalists take it off and they're using our predicted docking energies as ground truth. And that, of course, is not the case. Of course, we do the same with experimentalists. We use their highly noisy data and treat it as ground truth. But so I'd encourage you to call these things scores instead of energy. So even when you're trying to predict an energy, it's still a predicted energy. So I like score is unambiguous. So I'll be saying score. And why do I say this is because docking is designed to be high throughput. We want to screen. We're doing all the sampling, but we want to get an answer relatively fast. We don't want to wait a day, right, for each compound. And so we make a lot of fundamentally limiting approximations. The receptor is usually kept rigid, or at least mostly rigid. The ligand's usually flexible, but we limit it to like rotatable bonds. You're not like sampling bond lengths. And the big approximation is there's no explicit solvent model, right? Like we don't have water floating around, right? So I think with all these approximations, it's a real stretch to call this an actual free energy. Right, and the particular docking software I'm going to talk about is GNINA. There's, of course, many possible docking software, but what we work on is GNINA, and this has a lineage going back to Autodoc GNINA, which was an open source docking program that was designed from scratch by Dr. Oleg Trok at the Scripps Research Institute. It has no code in common at all with Autodoc. So this is an issue that sometimes people are a little sloppy. They shorten Autodoc GNINA to Autodoc, and that is incredibly confusing and wrong because Autodoc is a different docking program. So if you're going to shorten it to GNINA. And the focus in developing this is performance, right? He wanted to make it fast. He created a new really empirically driven scoring function that was optimized for pose prediction with affinity prediction sort of as second criteria. And this was published in 2009, but the last update to the software was 2011, and there's been no changes since then. Now, a while back, actually probably about, I think around 2011, we forked this to create SMINA, which stands for Scoring and Minimization with Autodoc GNINA. And so we were using it. GNINA is very nice source code. It's very well written. And so we were using it, but it wasn't doing everything we wanted. Specifically, we wanted energy minimization, where you can actually converge to the local optimum with respect to a scoring function. So we implemented that. And then we're doing scoring function development. So we wanted to make it easy to use different scoring functions. So we implemented that. And when I went to contribute it back to GNINA, they didn't want my changes. So we forked it, which is one of the advantages of open source software is you can do that sort of thing. And But part of the goal here is for the default with SMINA to basically be identical to VINA in its behavior, just to be easier to use. I say almost because we actually fixed a bug in VINA that has not been fixed in VINA with regards to optimizing hydrogens. But there's even a command line option where you can re-enable the bug to get exact identical output to VINA. So that's one of the goals here, is just a better VINA. It's an Apache GPL2 license. The reason we have the GPL, which is a copyleft license, is because we include Open Babel, which lets us support a much larger number of file formats. So it's very stable source code. It doesn't change much. It still is under development. We still update it if bugs come in or code bits rots. You have to prevent the rot, like it is operating systems are upgraded and new libraries come out. You often have to make changes to make sure things still build and work. And so we're still doing that with SMINA. And a lot of the features I'm going to talk about with GNINA are in SMINA because it has a subset of the features. But we wanted to keep this stable. And at the same time, we're developing these convolutional neural network scoring functions. And so we did another fork to create GNINA. So there's a fork of SMINA. And here we don't care about backwards compatibility anymore, right? Like in fact, our goal is to be better, for the output to be better than GNINA. The big additional thing in GNINA is the ability to do this convolutional neural network scoring, which involves pulling in a lot of other libraries and packages, especially CUDA, GPU optimization. So this makes it more difficult software to build and deploy, but it does give you the ability to do this CNN scoring. And so actually there's a preprint here, which was just accepted 2 days ago to the Journal of Informatics. So it's now an accepted paper where a lot of the data from this presentation is coming from. All right, so that's the lineage. Let's go ahead and go get the software. So if you haven't already, open this Colab link. You should see something like this and go ahead and just start running the first few cells. If you're not familiar with Colab, you just do Shift Enter to run a cell, and this is going to install the Open Babel software and Pi3D and what the wget lines here. Oh, I should say this Colab notebook's already set up to do this, but if you're doing this from scratch and you want to be running GNINA, you'll want to go to runtime and change the runtime type so that it has a GPU, because it is GPU accelerated. All right, so the wget, I'm just downloading the binary for SMINA. We're not going to use this, I'm just comparing. And here I'm downloading the binary for GNINA. And when you look at the size of these binaries, you can tell I wasn't kidding about how many extra dependencies GNINA has. And the reason is the easiest way I have found to distribute these is to package everything up into one, what I call semi-static binary, where most of the library dependencies are in there. Only like the sort of barest minimum of system libraries aren't included. And then and you build it so against older versions so that you can run on any reasonable Linux operating system. And then you can just download it and run it. And that mostly works. And so for GNINA, like this includes all of CUDA in the binary. So that's why it's so large. And this lets you get started quickly. You can just grab something from in Colab. But if you are running this a lot, you should just build it from source, and this will link it to the latest version of CUDA and all the other libraries on your system. And it won't be half a gig, it will just be 40 megs. So don't be too intimidated about this. This is just for quick getting to use it. So I see a question. I would have thought that Colab would be reproducible across everyone's instances. Maybe Drew, you could look into that. All right, once we've downloaded this, so also if for some reason Open Babel doesn't install in Colab, we can do a lot of things without it. So it's not the end of the world. Once you've downloaded executable, you have to actually make it executable. So this is just Linux for saying set the executable bit. Again, if you're not familiar with Jupyter, the exclamation mark is just saying run this as a shell command. So it's like you're typing it in the bash command line. And I can go ahead and run it. And now you have docking software installed. And so if you run it without any arguments, it tells you all the arguments it takes. And this is what we'll go over some of these and how to use this. All right, so here's the big question. Were you able to run GNINA and Colab? So oops. So I was. Oh, this is fine. This ran. Don't worry about this error message. So we have a standard for at least 30 people answering. All right, any questions to get the nose down to yes? All right, well, we've got a majority, so I'll keep going. But if, again, if you're running into issues, ask a question in the Q&A. All right, so but how does it all work before we even dock something? Well, this is the overview. You need to provide the inputs, obviously a ligand and a receptor, and also where do you want to put the receptor? So you have to define some box saying dock inside of this box. And then it samples conformations of the ligand with respect to the receptor and keep tracking the best ones using the scoring function. And then it refines, rescores, and filters those results to get the final output. And I should emphasize that all of this sampling, that like the actual core docking sampling was done by Oleg Trot. It's still from the same as in GNINA, where we've added things within the refinement and the scoring and the ability to introduce this convolutional neural network scoring at any point in this process. So let's set up example. This is all in that Colab. You should just be able to run these. I'm just going to grab a structure from the PDB. Then I grep out the atom fields, which gives me the protein. So this is the simplest way to prepare a protein structure. I'm going to sanitize it with OpenBabel because I don't want to keep seeing this warning, which is an okay warning. It's just OpenBabel has different opinions about where to put the double and single bonds in a benzene ring. And then this is pulling out the ligand. The name of the ligand is sp4. So I have created a separate receptor and ligand file. And here I'm going to look at them. And can you see the ligand I colored in green? So you really, I mean, you always have to look at your data no matter what you're doing in modeling. But here you would be checking the structure, looking for things like missing atoms, alternative residues. Are there cofactors, things that aren't protein, binding site waters? This is up to you. Do I keep them? Do I throw them out? Because I want to emphasize whatever you give to GNINA and say this is the receptor, it's going to say, okay, that's a receptor. right? So if there's a zinc ion on there, that's part of the receptor. If you have alternative residues where it's, you know, two versions of a lysine on top of each other because the crystallographer wasn't sure which one is right, it will say, okay, I have atoms on top of one another in my receptor, no problem. And it will just accept that. So it's up to you to make sure that the receptor structure is reasonable in what you want. You want to look for receptors that are already in a bound conformation. Rookie mistake is to forget to remove the ligand. It doesn't say, oh, this is protein, this is ligand. No, it says you gave me all these atoms, that's a receptor. So make sure you remove the ligand like I did before you dock. The big issue for protein preparation is protonation. Where are the hydrogens? So by default, open Babel is used to infer the protonation. So you give it a receptor with no hydrogens, whatever running this command outputs in terms, so the -H is adding hydrogens and the -XR is saying keep this thing rigid. If you look at what the hydrogens are in this file, that is what GNINA will be using. If you don't like that result, well, you can modify your structure. Since Open Babel will generally only adds things, if you put it in the hydrogen in a different place that still makes sense, it's not going to, you know, add surplus hydrogens. If you want to have full exact control over exactly where the hydrogens are, give a PDBQT. So if you give a PDBQT to GNINA, it accepts it as is, right? It will not try to process it with Open Babel. And so that is the way to have the most control over what your receptor looks like. And the hydrogens are important, obviously, but for determining, you know, donors and acceptors, because there's a hydrogen bonding term. For ligand preparation, any file format supported by Open Babel is acceptable, but it does need to be a valid 3D conformation. During docking, we sample rotatable bonds. We're not looking at bond lengths or angles, or importantly, ring conformations or stereoisomers. Those are not sampled. Whatever you give as input is going to stay the same, right? So you have a boat versus chair ring conformation, you know, whichever one you provide is not going to be changed. So if you want to sample these things, you will have to provide multiple versions of the ligand when you're docking. And just to emphasize this, only because I see this repeatedly, this is not a valid 3D conformation. This is a 2D conformation that has been laid out to be drawn in 2D. None of the bond lengths here are correct. But if you give it to GNINA, it will go ahead and dock it. You may even get a good score, but this is not a good starting ligand. You want a 3D conformation. Now, you don't have to worry too much about what this conformation is because we are sampling the rotatable bonds, right? But the actual bond angles and lengths need to be reasonable, and the protonation needs to be what you want it to be. Right, so this is this L3 file that I created is a generated conformation of the same ligand from that crystal structure. All right, so we have our ligand, we have a receptor. The final thing we have to define as input is where to put the ligand. What's the binding site? So this is one of the ways that SMINA and GNINA are easier to use than VINA, is a lot of times you have some definition of your binding site already, and it's defined using some molecular format like the bound ligand, right? You want a box around the bound ligand, or maybe you have the binding site residues, you want a box around the binding site residues. And so instead of having to extract a center and a size for the XYZ coordinates, which is kind of obnoxious, you can just provide what we call an autobox ligand with this autobox ligand command line option. You say, this is where the box is, and it draws a box exactly around that ligand. So whatever the minimum X coordinate and the maximum X coordinate is, that's the X range of your box. And then once it's drawn that box, it adds this autobox adds term, which is by default 4. So it's 4 on each side. So there's 4 here, 4 here, 4 here, 4 here. So it just extends it. And then there's a final check if you have a very long and thin ligand, and you create this box, you still have a long and thin box, and the ligand can't move around, and so you are really limiting your sampling. We then, by default, further extend the box enough to make sure that you can rotate the ligand. And so all those things happen automatically when you provide out-of-box ligand. And again, this out-of-box ligand, it doesn't even have to be a valid ligand. It just has to have atoms of some sort. So for example, if you've ever used F-pocket or MD-pocket, it produces this bunch of alpha spheres. So it's just a bunch of sort of atoms in space with no bonds saying this is where a pocket is. You can provide that as your out-of-box ligand. You can provide the binding site residues, whatever you want, as long as it has Cartesian coordinates. All right, so here's a thought question. Don't worry about getting right or wrong. What do you think happens to docking performance when auto-box add is increased? It's always a little tricky talking about docking performance because there's how well you dock and how fast you dock. So I'm asking about both. And in interest of fairness, I will say this is a trick question. Slower and worse. Let's see what people said. Slower but better, slower and worse. Not a lot of faster. So the trick question is because in terms of speed, it actually, for GNINA, it doesn't change anything. We don't, increasing the size of the box doesn't change the number of degrees of freedom, and it doesn't actually result in docking taking longer. So trick question, it shouldn't really matter too much. However, the quality is sort of interesting. So these are some graphs from the paper. So let me explain what you're seeing because we're going to look at several of these. This is looking at pose prediction performance. And so you do your docking and you get 9 results back, for example, 9 poses, and you say, okay, the top ranked pose, what % of the time is that predicted pose within 2 RMSD of the true pose, right? So here we're looking at 75% of the time, your top ranked pose is good, right? And then if you look at the top 2, it goes up, then top 3 and so on. Okay. And you can see the evaluation details in the paper, but basically this is looking at several thousand structures. And this is redocking on the left. So redocking is the really ideal, overly ideal case, right? You have the exact perfect protein that exactly matches your ligand. And in this case, you even have the perfect box, right? Because we're defining the box by the crystal ligand. For cross-docking, this is where you have 2 structures of the same protein. So structure A, structure B. You take your structure, the ligand out of structure A, and you dock it to structure B. And then you evaluate whether it's a good pose based on the alignment of structure A and structure B where the ligand is, right? So this is, cross-docking is more realistic evaluation. It's also, I should point out, not necessarily accomplishable, right? If there's a large degree of conformational change between the 2 structures, it's impossible to cross-dock. So you shouldn't expect to get 100% here. But this is what we're doing when we do docking prospectively, and it's a much harder problem. As you can see, we get down to like 40%. Right? And you see a big difference in the effect of auto-box add here. With redocking, the smaller the box, the better, because the box is exactly the right. You're applying a constraint that is the right constraint. You're reducing sampling to so that the right answer is more prevalent. And so smaller is just better. But this is very misleading because if you do cross-docking, a smaller box is actually worse because now the box is not the perfect constraint. So imagine you are defining your box using a small bound ligand, but the binding site is actually fairly large and can accommodate a much larger ligand, then your box is too small and you can't get the right answer. So the default's okay, but this is something you should be aware of. Again, if your box is, you have a small ligand and you want to dock larger things or larger binding site, you might want to increase autobox add. So it's really sort of a function of your system, what the right answer is. The default's okay, but again, think about how you're defining your box. Okay, so those are the three ways to specify input. So this is the minimal docking command. You should be able to run this in Colab. And if you go and run it, you should probably get slightly different answers, right? Because you'll notice it's using a random seed. Yeah, every time you run it, this random seed will be different. This is a stochastic process. The sampling is stochastic. Although at least for this system, every time I run it, you usually get a pretty good first pose, right, with a good score. Right? So that's the minimal command line. I would recommend a couple changes. One is I always try to set the random seed so there's reproducibility, so I can go back and get the same answer again. And it should be exactly the same answer if you're on the same system. You know, if you're on very different systems that might, I don't know, have different random number generators or something, then you might still get different answers, but on the same system, it should be the same. And the other thing I tend to forget to do is to say what the name of the output file would be. So the previous one tells you all these scores, but you don't get to see the poses because you didn't tell it where to put the output. And I will point out there's no problem with gzip files. You definitely want your output to be an sdf.gz, you know, save on disk space. And so that's all you really need to know to dock. So you can log out and go do something else right now if that's all you really want to know. But I'd like to dive into what's going on when we run this and how maybe we can use it for different approaches. But first, let's see how good the results are. So I'm going to measure the RMSD using the OBRMS tool from OpenBabel. I should point out on Colab, the package version of OpenBabel, I think, is OpenBabel 2. OpenBabel 3 is much better. We should all be moving to that if you're using OpenBabel. Slightly different interface here. You need another hyphen here. But this computes the RMSD between the first only here means the first ligand in this with respect to all the ligands in here. So these are my dock poses. So these are all the same ligand. And the nice thing about OBRMS is it computes the graph isomorphism between them. No, that's not a typo in Colab because Colab is using OpenBabel 2. Long story having to do with boost dependencies. It's only one hyphen with OpenBabel 2 and it's 2 hyphens in OpenBabel 3. But OBRMS computes this graph isomorphism. So like if you imagine you have a phenylalanine or a benzene ring, right, and computing the RMSD between this and this, where it's rotated 180 degrees. If you just say this is atom number 1 and you rotate it, then you think that RMSD isn't 0 because you've moved it. But in reality, it is 0 because it's symmetric, right? And OBRMS understands all that. You'll get the right RMSD. It's not doing one-to-one matching between atom numbers. It's actually looking at the graph. Okay, they're saying it does. Well, when I ran it, required one. Okay. But anyway, this is lesson 2 RMSD, so we're very happy. And so let's look at it. You always should look at the result. And this is cycling through them. And if we wait a little bit, it'll cycle back around to the first one, and you can see it has a very nice overlay. And actually, the second one, which had 4 RMSD, sort of just flipped this 180 degrees. Yeah, I will get to the CNN affinity. It's coming. All right, so here's the most interactive. Like in your Colab notebook, go ahead and write the command line for, we're going to dock a different structure file that's the same ligand, this L3 one, which we generated from a SMILE string, right? So There might be some concern that one reason that we're doing so well here is I am taking the crystal ligand and redocking it. So it's like all the bond lengths and angles are exactly right. You know, the things that aren't sampled are exactly right. So, but in prospective evaluation, what we're doing is we're generating conformers of new ligands, right? So what happens if we use a generated conformer? Okay, I'm told you do need 2 dashes for first only. I don't know why it didn't work for me. Well, that's almost a normal distribution. So if I do it and using this random seed, it actually gets a better score in terms of, so I am going to go into these more detail, but I will explain it now since there was a question. You know, this is the Venus score. So more negative is better. It's calibrated to look like an energy. This is a prediction of how good the pose is, and this is the prediction of the binding affinity. All right, so these are all good. And actually, when I do the RMSD, it's even better. So I mean, this is totally anecdotal, but you know, it doesn't, in this case at least, the reason we got a good pose wasn't because we're using the crystal ligand. All right, let me see this question. What if we do virtual screening? Would the final result be a sequence purely based on scores, or would each compound be separated with its own conformational scores? All right, so each, if you look at this file, it has 9 structures in it, each with its own score. Now you can filter that to just take the best score. Okay, so let's jump into what's happening here. Let's start with the sampling. So what are we sampling? We're sampling degrees of freedom. What are degrees of freedom? Well, first there's the 6 rigid body motions, translation, XYZ, and rotation, pitch, yaw, roll. And in addition to that, there's the internal torsions of the molecule. But again, not the other, we're not changing bond lengths or angles or ring conformations. Each one of these Monte Carlo chains starts by randomizing all degrees of freedom, right? So if you give it the crystal structure in the right pose, like all that information goes away, except for the bond lengths and angles, right? So there really isn't any bias to the starting conformation ligand other than the non-degree of freedom aspects of the conformation, like the ring pucker. Okay. So that's how all of these chains start. You have multiple one of these chains, so each one has a different random initialization. And then the way Monte Carlo works is you do some random move and then you evaluate it. And if the evaluation says this is a better structure, with some probability you move, you know, the better the structure is, the more likelihood you accept that structure, and that's your new basis for applying moves to, right? That's Monte Carlo. And so the moves we have are we pick a random transformation, translation, rotation, or torsion. You just pick one of those, randomly perturb it, and then that result, which is a random, you know, we've just random change, we then do a fast refinement. So we don't score it as is, we do refinement, which means energy minimizing with respect to our scoring function using a soft potential. So the by soft, like, because we did this random rotation or translation, there's a good chance that we're basically fully overlapping receptor atoms. And so if we use the scoring function as is, it would create this tremendous force that would just throw the ligand out of the pocket. And so we soften it so it just moves it a little bit. That's what we mean by that. And then use the metropolis criterion to accept whether to just sort of move along and keep sampling. And as you're doing that, you keep track of the best poses you've seen, right? And so the ones that you keep, by default, you keep track of the 50 best poses you've seen. So even if you move on to a different pose, if something's the top 50, you keep track of it. And that's the final output of this chain is here's the 50 best poses I've seen during this whole sampling process. And then you combine, you merge across all those chains, again, keeping the 50 best across those. And that's the result of sampling. There's a few ways you as a user can change it. The main way you should change it is with this exhaustiveness parameter, which is the number of chains. And what's great about this is all these chains are completely independent. So if you have 8 cores and you run 8 chains, they're all running on their own core. That's great. And so this is how I recommend increasing sampling. You get different random starts. It gives you the most oomph for the compute. In terms of how long the chain is, by default, this is actually set by heuristic, where it's longer the more degrees of freedom you have. You can set the number of steps, but that turns it from being a variable heuristic that depends on the degrees of freedom to a fixed number. And the only place this is possibly useful is if you're doing what I call a quick and dirty docking run, where essentially, for some reason, you want to generate a bunch of random poses, but you don't want them to be horrible poses that are, sterically overlapping horribly. And then the num MC saved is, you probably also don't need to change, but that's just how many best poses you keep. So if for some reason you want to generate a ton of poses, like more than 50, you might want to increase that. All right, so let's see how this works in terms of performance, how long it takes. So this is not running in Colab. I'll emphasize this is running on my workstation. If I run with exhaustiveness 1, it's, I don't know, 7 seconds. If I increase to exhaustiveness 4, it's actually still essentially 7 seconds. I mean, there's a little bit of overhead, but my workstation has 4 cores, so they're all running in parallel. So, it doesn't really hurt to have more exhaustiveness up to a point. However, if I tell GNINA to only use 1 core with this -- CPU one, then they don't run in parallel. So you can control the number of cores GNINA is using. And you can see, well, it's not 4 times, but it's definitely slower. And so this is something you should be cognizant, especially in a virtual screening exercise where you're going across a cluster and each node has so many cores, and you're going to want to control the exhaustiveness and number of requested cores appropriately. So ideally, you want the CPUs to equal the exhaustiveness unless you don't have that many cores. Okay. All right, so what's the effect of this exhaustiveness parameter? So the solid lines here are using our default scoring, and the dashed lines are using only GNINA empirical scoring. So like if you're using Autodoc GNINA. And one thing you'll notice is that default is much better. But we go to increase exhaustiveness, it's blue, black, red, and you can see what we see for a typical docking scenario is diminishing returns, in that the default of 8 is actually pretty good. And if you go from 8 to 16, that's twice as much computation, and you only get a little bit better. So I would generally stick with the default unless you have some reason not to. All right, which raises the question of the scoring. I just showed you GNINA versus the default scoring. So there's 2 kinds of scoring functions inside of GNINA. There is empirical scoring, which by default, we're still using the GNINA scoring function. These are fast, interpretable scoring functions. Yes, GNINA also splits Monte Carlo chains across cores. There's a question there. And so does GNINA. Like this this whole part, that's all Oleg Trot. He implemented that. Okay. So these empirical, you've seen scoring functions like this. You've seen force field. It's a bunch of terms, most of which are pairwise interactions. They have some weight on them, and you sum them up, right? And so you get some score, and you can say this amount of the score came from this term, that term, that term, right? So very interpretable. So by default, these are used for this fast refinement during the search. They are even used for this full refinement at the end because they're fast and they work well. Then there's this convolutional neural network scoring, which is much slower if you don't have a GPU, but is more predictive. Like that is the difference between these dashed lines and the solid lines is the convolutional neural network scoring. So you're getting more than 10% increase in success. And by default, it's only used for this final ranking at the end. So you come up with these 50 best poses, and you re-rank them using the CNN scoring. So it's just at the end right here. And I should say there's a final step here. So you re-rank them, and then you go through with an RSC filter. So like if the top 3 are all within 0.2 RSC of one another, you only keep the first one. Right? So you get some nice selection, a diverse selection of poses. All right, so let's dive into this empirical scoring. Autodoc-VINA scoring, this is this exact same term as Autodoc-VINA. This is from their paper, and it really shows the scoring function quite well. There's really only 3 kinds of potentials. If you have 2 atoms next to each other, they'll have a potential like this, kind of a van der Waals with this interesting little Gaussian out here. So at this point, the 0 is where the atoms are touching, where their radii, match. And so that's the optimal place. If both atoms are hydrophobic, then you're even happier to have them touching. So that's this red curve. And then if they form a hydrogen bond, you actually want them overlapping a little bit, right? Hydrogen bond, the atoms get closer, and that has a more effect. So you actually have this curve like this. And yes, there is this discontinuity here, and it goes like that, right? This is the Autodoc-VINA. There are no electrostatic terms. There's no partial charges. I emphasize this because this question comes up a lot. People think partial charges matter for VINA. It does not. All it cares about is this a donor or an acceptor, and whether something is +2 or just happens to be a donor, it's going to have the same potential here. There's no explicit metal term, but metals are modeled as hydrogen donors. Where did these terms come from? Many years ago, I asked this question on a internet bulletin board, discussion board that's no longer in existence, and I was assured that a paper was forthcoming that would just answer it in detail, and that paper has never come. But my understanding is there's basically a massive grid search across all possible terms, like many terms, electrostatic terms, solvation terms, and with a focus on optimizing for pose prediction quality during redocking. And then once they found the terms that work for pose prediction, the final scoring function was linearly reweighted so that the output fit free energy, so it had the right sort of scaling, so that you would have these free energy units. So that's my understanding where that came from. And so if I run with a higher verbosity, it'll actually print out what the scoring function is. So here's those same terms I showed on the graph. One additional thing here is there's a non-pairwise potential. There's a count of the number of rotatable bonds. So you're penalized for having more rotatable bonds, is a ligand term. And so the default's VINA, but we have other built-in empirical scoring. Probably the most interesting is this Vinardo potential, which is described in this paper, a totally separate group. where they went in, they took GNINA, they said, okay, this is fine. We're going to reparameterize it. So not only are there different weights on each one of those terms, they optimize the radii of all the atoms. So it has different radii for each atoms, and they got better results. I'll say in my hands, it's a little mixed, which one's better or not. Although I'm going to show you some results where it does appear to be better. So you might want to consider using this instead. We also have an implementation of AutoDoc4 scoring. And all the other built-ins one you should just ignore. They're sort of in there for posterity. They were talked about in papers. They aren't very good. Okay. What if you want to create your own scoring function? You can do that, and you can do that easily as long as it's a collection of terms that are already implemented. So here's all the sort of different terms that you can apply. So all these ones down here are properties of the whole ligand. But these are pairwise potentials, and the signature here matches the function, and so they're parameterized differently. So if I'll just look at a Gaussian term, you have your offset, so 0 means the atoms are touching. The bottom, the Gaussian will be when the atoms are touching, and the W is the width, and for all of these, the C is the cutoff value, right? And so you can dive into these and parameterize them how they want, and you have Leonard-Jones and quadratic terms and hydrogen bonds and all those things. So the way you provide this to GNINA is with a text file where the first column is the weight of the term, and then the parameterized term where I actually specify, for example, for the Gaussian, the offset should be 0 and the width should be 0.5, right? With a cutoff of 8, right? And then it'll just apply this for you. So here's an example. If I give it with custom scoring, everything.txt, it will print out, and running score only, it prints out conveniently these. If you grep out for these hashtags, you basically have a CSV, well, a space separated file where these are the column names, and then these are the values there for each term. So then you can use this, and I'll show how to use this in hopefully we'll have time at the end of the presentation. So this is to help you develop your own scoring functions, or maybe learning a QSAR model using structure interaction features. Just one super hacky use case, which is what I call soft covalent docking. So all of this is the standard GNINA scoring function. And then we add in a Gaussian that says, I want chlorine and sulfur to be, like, I really want, -100, I really want chlorine and sulfur to be next to each other. And then I modified my system so that there was a sulfur on the receptor and a chlorine on the ligand, and they happen to be the only chlorine and sulfur in the system. And so that sort of forced them to be next to each other. And this is what I call soft covalent docking, because you aren't forcing there to be a bond there, you're just really preferring there to be a bond. But it's also really hacky because they had to, make non-physical changes. It wasn't really a chlorine. If you're lucky, you have a system where maybe you want nitrogen to coordinate with an iron, and there's only one iron in the receptor, and you can just create a nitrogen iron term like this, and you can go and it will prefer poses where the nitrogen is next to iron. So a goal for a future version of GNINA is to make covalent docking much easier and not hacky, but this would be one way to do that. So that was the empirical scoring. The new fancy part about GNINA is this deep learning, and this is where we use convolutional neural network scoring. So a convolutional neural network is very popular in image recognition, and the idea is you pass this filter, which is a learned function over this image to create feature maps. And so it might be learning, here's horizontal lines, here's vertical lines, and storing that information in this feature map. And then you do this over and over again. So you learn higher and higher order features, like from lines to corners to shapes to animals. And what we do is we take a 3D representation, so basically a 3D picture of this protein ligand, where the atoms are represented as Gaussian densities on 24 angstrom grid. And I'm bolding that because it's important. If your ligand is bigger than 24 angstroms, you're going to see weirdness happen. So that is a limitation of GNINA. And then we push it through convolutional layers to produce two numbers. One is the pose score. How likely is it this is the right pose? It's within 2 RMSD of the true pose. And the other one's affinity. What's the predicted binding affinity? And we have we have several publications on this. I don't really want to dive into the machine learning aspect, but there's a lot of models that are built into GNINA. This dense model is the biggest one. It has 3 dense blocks like this, which each have 4 convolutional layers. And they all take this 3D representation and output this affinity and pose. And the default actually isn't to use just one model, but to use an ensemble of models, because you get a nice improvement in performance when you use an ensemble across these different models trained on different data sets for a more robust result. And so you can see here's all of our models, and by default it picks 5 of these. And so we did this sort of greedy selection algorithm to find sort of the 5 models that gave the best performance, and while still fitting within a reasonable amount of GPU memory. And so, like I said, this predicts the pose quality, the CNN score, and the binding affinity. And so this is just showing them. So this score is going to be from 0 to 1. You can think of it as a probability that this is a good pose. And this is an infinity score. It's in PK units. So this is trained on all sorts of binding affinity data. But roughly a 6 would be saying 1 micromolar, and a 9 would be saying 1 nanomolar. You shouldn't take that as like a truth. It's more useful for ranking. And then finally, if you have an ensemble, you'll see this variance, which is telling you how much the different models in that ensemble disagree. So you want this to be lower. Like the more confident the ensemble is, the lower that will be. And so just to compare the black here is the default, that default ensemble, which you get for free. You don't have to specify any different options. And then these are just looking at individual models by themselves. And GNINA is in blue. And I mean, the main thing to note here is, we're significantly better than GNINA. We're also significantly better than our, like the first model we published back in 2017, which is the yellow. We haven't solved the problem, but we're definitely making improvements. Let me just remember where I'm at, because I do want to take a break shortly, but I'm just going to do a few more minutes. All right, so that's the scoring. And then the final step is this ranking and filtering. So again, like we have 50 poses come out here. We score them and rank them. So by default, we rank by CNN score. We want the most likely good poses first. You can adjust this with the sort order to pick different ranking criteria. Be aware that so if you're outputting 9 poses and you change the sort order, it's not going to be the same 9 poses, just in different orderings, because what you're ranking is these 50 poses coming in. So a different number, and there might be a different top 9. And then by default, anything that's within 1 angstrom RMSD of a better ranked pose, we just don't output. So you get a nice diversity of outputs. And so we only do the CNN scoring at this very end. And the reason we do that, you can use CNN scoring for refinement, for actually using the CNN score itself to optimize the ligand. But we did not find, and I actually find this very depressing, is we spent a lot of time developing these CNN scoring functions. We didn't find that refinement helped. Like these black lines are on top of each other. So that's depressing, because I would love it if the CNN scoring had this much better energy landscape that let you optimize ligands to the right answer more easily. But the silver lining is refinement is much slower because you have to keep applying the CNN scoring. So the fact that rescoring actually works well in terms of docking performance. And just to emphasize this, though, you really want a GPU here. Running with a GPU, the full docking takes 11 seconds here. running without a GPU, which is what this is doing, this took over 2 minutes, right? So you can dock without a GPU. You can, if you, and it might even make sense if you have thousands of cores versus just, a few GPUs, but it is much slower. And you do need an NVIDIA GPU. You probably want at least 4 gigs of RAM. It's always better to have more. Okay, I'm going to pause there because it's 10 o'clock. And what do you say, 5 minutes?
Yeah, 5 minutes, I think, just to let people to have a stretch their legs and grab a cup of tea or something.
Yeah.
Tammy, are you online?
Hi, can you hear me?
Yeah.
Yeah.
Do you want to just say a few words?
Yeah, thanks.
Tammy's from Liverpool ChiroChem.
Thank you, Chris, and thanks, David, for the excellent workshop. So as Chris said, I work with Liverpool ChiroChem, and we are a chemical technology innovator on a mission to accelerate small molecule drug discovery. We are sponsoring these events because we understand the importance of these open source computational tools to the global research community. So we would love to support their development and spread awareness about LCC's chemical space at the same time. At LCC, we are committed to providing chirally pure fragments and drug-like sized molecules for screening. As we know, binding sites are 3-dimensional, and so understanding the stereochemistry of molecules for screening is vital. If you would like to find out more or to discuss how LCC can support your projects, please get in touch on our LinkedIn page at Liverpool ChiroChem or by emailing sales@liverpoolchirochem.com. Thank you and enjoy the rest of the workshop.
Okay, thanks very much, Tammy.
That's all right.
Okay, we'll be back in 5 minutes then. Okay. Okay, David, you want to start?
Yeah. So there's a question about accuracy. Binding affinity prediction is really hard. If you look at our crosstalk paper, we do assess that, and it correlates, but this is not like a free energy calculation, like I said at the beginning. Okay, so we went through the whole docking workflow. I want to address a few common sort of cases besides just targeting a binding site. One is whole protein docking. So this is actually really easy to do. All you do is you set your autobox ligand to be the receptor, and it creates a box around the whole protein, and you're done. And so if we do this, actually, it works for this example, it works really well. It's going through the different, you can see it's sampling different all over the protein, but actually the first two, if it cycles back around here, are right on top of the right post. Like this is redocking. Like I said, that's an ideal case, but it does actually find the binding site. And in general, of course, whole protein docking, you're not going to do as well is defining the binding site. But this is the results here. Again, the dashed lines are VINA versus using the default with the CNN scoring is the solid, which you can see we do better with CNN. And unlike when you have a targeted binding site where my recommendation is to sort of leave the default exhaustiveness, what we found with whole protein docking, not too surprisingly, is we don't see really diminishing returns. Like this is a much larger search space. So just keep increasing exhaustiveness is to the limit of your patience, and you'll probably get better results. Right? As we keep increasing it, you keep getting better results. One thing I like, if you look at this, you compare the CNN scoring to just using default VINA. Even the default exhaustiveness here does as well as VINA with 64 exhaustiveness. So even though you spent 8 times as much time searching with VINA, you get the same result as with just the default amount with the CNN rescoring. So I think it really shows that even though the scoring is slower, it can actually be faster overall when you look in the context of something like this. So, but the main recommendation here is whole protein docking, crank up that exhaustiveness. Flexible docking. So this is actually something Rocco worked on a lot. So feel free to ask questions and he can answer them. So with GNINA and SMINA and VINA, side chains can be treated flexibly. The backbone is always rigid. We're only sampling the side chain conformations. So here I'm just setting up an example, which is just an alternative protein for the same kinase. This is just setting up the example. You should be able to run this in Colab. And so let's do a cross-docking where we dock the ligand from 3 ERK to the 4 ERK structure. So you see our receptor, so the REC2 is the 4 ERK. I'm docking the original ligand. I am specifying the binding site using the ligand from this receptor. This is how you do cross-docking. You know, in prospectively, you don't know what the perfect box is for the ligand you're docking, right? You have to use the known binding ligand. And then so I output it. And you know, if you remember what the scores were for the previous exercises, these aren't nearly as good. In fact, when I look at the RMSD, you know, these are pretty bad. So it didn't work very well. And if we look at the results here, so this is a little bit confusing. So 3 ERK here is green. This is where I'm getting the ligand from. 4 ERK is yellow. And if you look at this binding site, you can see there is a fair amount of, if you compare the yellow and the green side chains, there's several that are quite different in conformation, right? So you might suppose that flexible docking would be helpful here. Right, so you might want to try this. So how do we specify what side chains should be flexible? So you can provide a PDBQT file that says exactly these are the flexible side chains. This is a real pain to set up because you have your flexible part of the receptor and your rigid part, and you have to divide them into two different PDBQT files. That was a real pain. That's how VINA did it. So we have this ability to basically just say, here's the flexible residues. And this is what I recommend is you spend time looking at your structure, thinking about the minimum set of side chains you want to be flexible and manually specifying. You say chain A, residue 300, right? And it will do the rest. That requires manual involvement in thinking about the structure. If you're doing a systematic evaluation or if you're just lazy, you can also just specify in terms of distance from a ligand. So in the case of like this 4 ERK, you have the bound ligand and you can say, okay, any residue that's making contact that's within 4 angstroms, make that flexible, right? And so you have to specify the ligand and that distance, that 4 angstroms, 3 angstroms, like at what threshold do I want to say this, if there's an atom in the side chain, it should be flexible. Then these other two, if you're doing this in sort of a systematic evaluation across many PDBs, as Rocco knows, like sometimes you have so many things flexible, the docking takes forever and you don't want that. So these two are ways to limit, like only take the smallest number of, a smaller number of side chains. So you limit how many flexible residues there are, either a hard limit, meaning it won't even dock if it exceeds that limit, or a soft limit where it just takes, you know, the end closest residues. And finally, you know, all this work of sampling side chains, you might actually want to see the results of sampling, so you can output that as a separate file. Now, because you know, if you're you have all these different residues and the whole rest of the protein isn't changing, so it would be a little inefficient to write out an entire protein where most of the atoms aren't changing. So we only output the flexible changing side chains. And then if you want to reassemble that into a whole protein, we provide a script that will reassemble that for you. So here's an example. So we're going to be lazy. We're going to take, oh, this is a typo. This shouldn't be 3, it's 4, right? So the flex dist here is 4, and the ligand is the cognate ligand. And I go ahead and post. And if you run this, running this in Colab, you're waiting now. This takes much longer. But you notice the scores actually are better now. So maybe we actually were successful here. Let's see, there's a question. What is a recommended max number of residues to make flexible for reasonable docking times? 0. I'll stick by that answer. Actually, so we do have, before it was, I don't know, 7 RMSD was the best post, and now we're, under 3 at least. Although when you look at this, sort of the functional groups are oriented in the right spot, but it's not really that great. This is flexible docking's hard. Now, if I spend some time looking at the structure and playing around with it and thinking about it and cheating and evaluating different docking runs, I can do pretty well with just 2 flexible residues as long as I increase the exhaustiveness. And actually, with flexible docking, increasing the exhaustiveness is something you might want to consider. And so here, if I just look at the RMSDs, I did a little bit better. It's not gangbusters, but this runs much faster than the other one. And so part of the way I chose these residues is I had both structures, which is kind of cheating. And I said, well, here's the residues I'm marking flexible here. And you notice it prints out and it tells you which residues are flexible. But some of these, and like comparing the structures, like they aren't really moving between the 2 structures. So why do I want to make these flexible? Anyway, you may thought I was getting a facetious answer when I said 0, but really that is my recommendation, is to not do flexible docking. And so at least for GNINA, I'm not making this claim for flexible docking in general and all docking software, but this is an evaluation Rocco did. And he docked to the rigid receptor and he docked to flexible using, I'd have to check the paper, but residues within the ligand, using this FlexDis thing, and compared what's the difference in RMSD with not rigid versus flexible. And so anything that's to the left here, you actually got a better pose doing rigid docking than with flexible docking. And he binned these by how far apart the binding sites were, right? The binding site RMSD, like how much is it changing, right? So yes, when the binding site is changing a lot, then flexible docking, you get over to the right here. But one thing that isn't showing here, but there's another figure in the paper that shows it, is most of the structures we look at, if you just sort of pick 2 structures of a protein from the PDB, the differences are more down here. All right, so this is why I feel that if you're doing flexible docking, it should be guided by some additional knowledge that you have. Like you've looked at the protein structure, you see there's this arginine that is, at the lip of the binding pocket that can clearly float around. Like you sort of have a good reason to say this should be flexible, because when you make things flexible, you're increasing the number of degrees of freedom. You're making sampling harder. You're getting providing more chances for scoring and sampling to get things wrong. So by default, I would actually recommend rigid. The other thing I would recommend is, instead of doing the flexible sampling here, is docking to ensemble conformations, because if you generate if you have an ensemble from NMR or from just multiple structures in the PDB, you're also getting the backbone flexibility there, which we can't do in GNINA. So that's my recommendations for the current version of the code. All right, virtual screening. Of course, a lot of times we ideally would like to be able to virtual screen. So this is unpublished, but hopefully not for too long, results where we evaluated on the DUDE directory of useful decoys virtual screening benchmark. Now, there are a lot of issues with DUDE, especially for training machine learning models. The thing I'd like to emphasize here is we did not train on DUDE, right? Like this is just using the built-in default scoring. which was training for pose prediction and binding affinity prediction, was not training for virtual screening. We weren't using, very little overlap in that training set with DUDE. Having said that, there are sort of biases that exist in all data sets. However, with those qualifications, we still are on average doing better using GNINA than using just pure VINA or even Bernardo. So each dot here is a different target in the DUDE set. So there's 100 different targets, 102. And so you do and there's actives and decoys, and you dock them all to the binding site. There's a reference receptor, and you compute an area under the ROC curve, right? So 1.0 means you put all of the actives first, rank them all first, and all the inactives last. And 0.5, which is a dashed line, means you're doing random, right? And you say most of the cases were above random at least. And if you just look at the medians here, Bernardo is actually doing better than VINA. Our default is doing better than Bernardo. And a consensus, and I'll show you how I did the consensus in a little bit here, where I combine Bernardo and the default does even better. And what I'm ranking here, by the way, so I picked the pose using the CNN score, because this is saying this is not what I think is the best pose. But for ranking for virtual screening, I'm using the CNN affinity. I'm saying this is the highest affinity. So this is AUC. Another way to look at this enrichment factor in the top 1%, and it's the same trend here. And so we do see an improvement. All right, so having seen that, you're like, well, let's go dock a library of compounds. Let's see how we do. And so here's my recommendations for that. So first of all, you want to pre-filter your library, right? Because docking is expensive. You don't want to waste time on crappy ligands. You probably want to remove highly flexible ligands because they will take a lot longer to dock. So, diminishing terms in terms of, likely of getting something good versus the amount of CPU time spent. When deploying this across a cluster, watch your CPU usage. You want to explicitly set the number of CPUs, the number of cores, it's called CPU, but the number of cores that GNINA is going to use, and make sure this is less than or equal to exhaustiveness, right? One thing I have seen happen on our own cluster is people run it, they don't set the CPUs, and they submit a whole bunch of jobs, and now you have 64 GNINA jobs running, all using all the cores. It's not good. It's okay to share a GPU because by default, this GPU is only being used in that rescoring, right? So it's not being used across the whole docking. So it's okay to share. But by sharing it, you need more memory because every GNINA program instance will need GPU memory. So you may be limited that way. Like if you have 64 cores, you might not actually be able to run 8 GNINA jobs if you only have a 8 gig GPU card. Avoid unnecessary receptor processing, right? So one run of GNINA is going to read in the receptor and process it, and it's going to read in ligands and process it. You can't, but you can't get around that. But it's the same receptor every time, so you don't want to do redundant work. So one way to reduce the amount of receptor processing is pre-process it to a PDBQT. So then again, it skips the open babel analysis, so it's faster. Even better is don't have like one job per ligand, right? You don't want to launch a million GNINA jobs to dock a million ligands. You can give is your input ligand a multi-ligand file. It can have 1,000 ligands in it. And so it'll pre-process the receptor and they'll just go through ligand. And so you amortize the cost of pre-processing and starting up the application and all that stuff. So you really want to split up your library into large chunks to dock. And my final recommendation with high throughput screening is to not do it, at least with docking. It's really computationally intensive. There's better reasons to burn CPU and GPU cycles, in my opinion. So what's the alternative? There's a certain amount of self-promotion here, you should be aware, but the alternative is Pharmaco4 search. So the advantage to this is you are using your expert human insight to define a query. Like you can say, this is what I want the molecule to look like. And it's really fast. You can screen millions of molecules in seconds. The results are already docked because they're aligned to the Pharmaco4. And you can still get that last bit of docking, that refinement and rescoring. You can take those Pharmaco4 aligned poses and refine and rescore them with GNINA and still get all of this benefit. The big disadvantage is it uses expert human insight to define a query, right? You have to get the right query. You can't just sort of blindly say, here's a protein, you figure it out. And this is especially difficult when you don't have a bound ligand showing you what the potential interactions are. And if you want to explore different interactions, you have to have multiple queries. All right, so here's my question for you. Let's see how many people are still with us mentally. Have you ever done a Pharmaco4 search? And specifically, if you've ever used Pharmit. Can we get up to 10 this late stage? No. Well, this is your lucky day. Well, I have. Okay. Oh, wow. Good. Well, presumably you're interested in screening, so you're going to have a new experience today. All right, so we've developed this software. It's a Pharmit called Interactive Exploration in Chemical Space for doing Pharmaco4 search online. And I'm going to demo it now. And I'll start with an example. Uh-oh. This is always dangerous. All right, so this is just one server, so don't repeat what I'm doing because you'll just nail the server. And what we're doing, or what I did, this is estrogen receptor alpha. It's a bound crystal. And we have a Pharmaco4 here, which are the essential features in the interaction. It's a 3D Pharmaco4, so it includes their spatial arrangement. And estrogen receptor has this really well developed and understood Pharmaco4 where you have this linear hydrophobic core with these two polar ends. And you can use this to screen, right? So what you're looking for are compounds that match all these constraints, that if we just look at this, that you have a hydrogen acceptor, orange is hydrogen acceptor here, donor acceptor here, and hydrophobic points features all through here, right? And that took, what did I screen here? I screened Molport, which is a aggregator of commercially available libraries. So if we look at, so 7.8 million molecules, and we screened that in a few seconds. And these are all things that exactly match this Pharmaco4 query. And there's a lot of other libraries in here. I think maybe I need to talk to our sponsor to see if we can get their library in here, that you can screen with millions of molecules, 126 million molecules from MKIL, 45 million here. And you can do this in interactive time. So you can play around with these, the query and look at the molecules you're getting. But what I'm going to do now, and here you can do this too, is I'm going to go to these contributed libraries because you can submit your own libraries to screen. And I'm going to look for benchmark and pull out the ER alpha benchmark. And so this one, it should be okay if everyone does it because This is from DUDE. It's the ER alpha benchmark. So they have some active ligands and some decoy ligands, and it's does it say how big it is? It's much smaller than millions of molecules. So I think we should all be able to screen this one. So again, you go here, you go to contributed, just search for benchmark, and you should see DUDE ER alpha benchmark, and select that one. And I can screen for all the compounds in this small library that match this Pharmaco4. And one thing you'll notice, the reason this is called the benchmark is the compounds have been annotated with whether they're active or not. And you notice I'm getting a lot of active compounds. This is actually, like I said, this is a very well-developed Pharmaco4. It works well. And because they've been annotated, I can compute an enrichment factor and an F1 score. So the enrichment factor means this is like 55 times more enriched than if you just randomly sampled this number of compounds. The F1 score is the geometric mean of precision in the recall. And you'll notice it's actually pretty low, which might be surprising because if you look at these are pretty much all active. So I have a super high precision. I think it's 1.0 precision, it looks like, because everything is active. But the fact that this is low is telling me that I'm missing a lot of compounds. I have a low recall, right? So this is something I want to optimize. Like I'm only getting a small percentage of the actives that are actually there. And I can do that by editing the query in real time. So I can just disable some of these hydrophobes. Maybe I'll keep the ring, but not, I don't know. I forget what I did before. So feel free to play around with this. The other thing I'm going to do, and I actually recommend always doing this, is I'm just going to set the hydrogen bonds to 1. I kind of regret making the default be 0.5, but it was really only to do that so that it was easier to distinguish the spheres. And so by increasing this radius, what this is saying is the region where you could have an acceptor, right? So by making it bigger, I should get more hits. So here I have 59 hits. If I search, why did it change to Campbell 925? How'd that happen? Always dangerous to do a live demo. So here I have 1600 hits. So I made the query less specific. I still have, like, if I just look at the top ones here, they're all active. My F1 went up really much higher to 0.46. I still have a pretty good enrichment factor. And you can play around with this. Let me just, I think we have enough time. So let me go through this. And if anyone has any questions, please ask. So these are your Pharmaco4 constraints: hydrogen bond donors, acceptors, hydrophobes, aromatic, positive, negative charges. You can, for aromatics and hydrogen acceptors, you can specify directionality if you want. I generally don't recommend it. It's sort of it's an additional constraint that isn't always necessarily right. And so that is saying this is what this molecule must have. It must have these features in these positions, right? But it's not saying what the molecule can't have, right? Like there's no way to say don't have a hydrogen donor here. But one way we can filter the molecules is by shape. Like you could just say I want there to be something, anything here. But more commonly, you don't want your molecule to overlap the receptor, right? So you can define an exclusive shape. You can use spheres, but we can just use the receptor, and I'll increase the tolerance here. And oh, there was a problem with Chrome. This was the problem with Chrome. It does not happen in Firefox. But basically, where it's blocking out the region where the receptor is and saying you shouldn't have anything overlapping there. I had forgotten about this issue. and that will filter out those overlapping molecules. And I'll just reload. What the? This is the perils of a live demo. Let's see what some of the questions are. Pharma industry people using these tools and reporting success for pharma? I know people are using it. In terms of reporting success, there are publications using it. They're not. I haven't seen anyone from like a big pharma really impressive publication. Unfortunately, a lot of them tend to be these made a model and screened it sort of publications, which is a little disappointing sometimes. But people are definitely, I mean, we are using it in getting hits, and there are publications where people are getting hits. Have you got benchmark scores on Dude using pH4 versus GNINA? When you say pH4, I'm thinking of the MOE Pharmaco4 format, and I'm guessing that's not what you mean, so I'm not sure. So well, you need to. This is the issue with Pharmaco4 search. You have to define the query, right? And if you just take the default Pharmaco4 that you get from the bound ligand, often it's too specific to be useful as a query. So it's not clear how to benchmark this because part of what you're benchmarking isn't the search process, it's the query definition process, which is something we're interested in looking at as well. So this is a fully enclosed binding site. So you just see a bunch of blocks here, but I'm going to increase the tolerance to sort of 1.5. And what this is saying is it's going to filter out anything where the atom centers overlap, are sort of more than 1.5 angstroms embedded into the protein. And let's get rid of a few of these. This is fun, right? You should be following along. So now there's probably there's fewer overlapping compounds. Let's see, what else can we do? Just checking the time. Yeah, so there's sort of the normal visualization things. You can have a surface, but this is sort of inside the protein, so that's not very useful. For screening, there's all the filters here. I actually don't. My advice is to do minimal screening here. So with docking, you want to screen as much as possible up front because it's expensive to do the docking. It's not expensive to do the search. So to me, it makes sense to wait till the end where you already sort of have more information about like what is the score, you know, what are the interactions, what's the ligand look like before you start throwing things out. And so I'd actually just recommend leaving these. One thing to point out is what are we searching here? We're searching pre-generated rigid conformations of the ligand. So you actually get multiple hits per each ligand. So if you only want, you know, one conformation, you can set that here, and you'll have fewer hits, and you just get the best conformation. Finally, this is just matching a Pharmaco4. We need to rank this, and you can do that. You just click this minimize button. And what this is doing is applying GNINA, actually, is in its optimizing, so rigid receptor, but flexible ligand. It's actually now moving the ligand and changing its torsions to optimize for the GNINA scoring function. And that gives you a score that lets you actually rank these compounds. And in addition to this score, there is this mRMSD. This is how much the ligand has moved from the Pharmaco4 aligned pose to the energy optimized pose. And the thought here is you came up with this Pharmaco4 for a reason. If the ligand has moved a lot, even if the score is good, you probably don't want that ligand. You want it to be making those interactions that you asked for. And so you can filter by the mRMSD. And I would recommend doing that unless you don't have a lot of confidence in your Pharmaco4. Right. Now, this is only GNINA. We don't have the CNN scoring yet here. Is there benefit to you? Yeah, so that great question. So this is we don't have that CNN scoring, which I showed you had better post prediction, better virtual screening, right? So how do we get that in? Well, at the moment, you need to download your hits here, which, if you can do this. Oh, really? Seriously? And that's where we're going back here. So you've done all this screen, you've interactively optimized your Pharmaco4 some way to get a good number of hits. What I usually tell my students is, look for somewhere between 1,000 and 10,000 hits, just sort of a rule of thumb to know that you don't have something overly specific that's still manageable. And then you have these that have already been minimized with GNINA, but we want to rescore them. Oh, right. So just to emphasize what I've already said, Pharmit lets you search all these rigid conformers really quickly, millions of compounds. You can also upload your own libraries, both public and private. And this query says what must be present. So there's still more room for refining. Just because it matches the query isn't enough to really do the full virtual screen. Yeah, I said all this. So this was, I applied this Pharmaco4 and I did this search and got these results, which was similar to what I think I did. And I downloaded them. And now I'm going to rescore them. So this is where you get the CNN. This is where you get to use the GNINA as part of this Pharmaco4 search process. So this is 4PPS is the estrogen receptor. So I'm going to grab that. I'm just going to pull out the protein. I just do that, but you got to look at your structure and check for like these issues with missing residues and so on, right? And then minimized results is just a file I downloaded from Pharmit. I give it my receptor, and I say -- minimize. Now this isn't actually strictly needed because they're already minimized, but if needed, I'll do a further refinement. And here I'm going, instead of using GNINA, because they already got these already minimized with GNINA, I'm going to minimize them with Bernardo, right? Since it seemed to do a little bit better. So they were GNINA minimized. I'm going to minimize them with Bernardo and score them with Bernardo. So here it's printing out. This affinity is now the Bernardo score, not GNINA. This RMSD is how much it moved during the minimization. So these are Bernardo and GNINA are pretty similar, so it's not moving a whole lot. And then I have my CNN scores again. All right, so all of this is printing to the screen, but all this information is also in the SDF file, is SD data, right? So if you look at the file, there's these tags here with all this information, and so you can read it in. And so here I'm just going to use pandas in Open Babel. So I'm reading in the file, and I'm getting the name of the molecule. And then with PyBel, you get to the SD data with mol.data. And so I can just pull all of this information out. And now I have a pandas array that has the molecule name and then the 3 different scores now, right? Because I have the CNN score, affinity, and Bernardo. And this label is true if the word active is in the name, because this was a benchmark set, right? I actually know the right answers here. And so this lets me evaluate the different scoring. So I can do that. So here's an ROC curve, so false positive rate over true positive. You want to go straight up and then over. Right, so this is performance on one target. And if I look at Bernardo, actually the AUC is worse than the CNN affinity. But if I look at what's happening early on, and this is one reason why it's always good to look at ROC curves, right? Because the AUC is giving you sort of this overall statistic that maybe isn't what you really care about. Bernardo is actually better here because it's almost 20% of the true ligands it is just putting right at the front with no false positives, right? And for virtual screening, that early enrichment is really what you care about, because you're not evaluating 10% of your library. You're just going to evaluate the top hits. Now, on average, I did show you the EF 1% numbers. On average, CNN Infinity still does better for early enrichment. But for this one target, Bernardo is doing better. They do the CNN Infinity, it's tiny, it gets a few, it goes up a little bit, but then, it's almost random performance until you get to, I don't know, here. But this is why I recommend for virtual screening, developing a consensus approach. Especially when you don't have a benchmark like this where you can actually see what the right answer is, you want to sort of hedge your bets and combine them. And the way we do this is actually very simple. Is we take the rank produced by CNN Infinity and the rank produced by Bernardo. I'm taking the negative because more negative is better with Bernardo. And I'm just taking the sum rank, right? And actually just these sort of min rank, sum rank, consensus schemes seem to work really well. The advantage of doing the rank is you don't have to worry about differences of scales or units, right? It's like this one is number one, this one's number one, and you just if you have a ligand that is highly ranked by both scoring functions, that's a really good thing. If the same ligand is number one with both scoring functions, that's great, right? But if there's a lot of divergence about where it is in the ranking, then maybe it's not so great. And so you can see with this consensus, you get pretty good early enrichment and you get the good overall AUC. And so this is exactly the sort of consensus ranking I did when I showed you those virtual screening results, where the consensus was even better than just CNN Infinity or Bernardo by itself. If you have access to other docking tools or other scoring functions, I think it's a great idea to evaluate them all and look at a consensus across all of them, right? Because if you have ligands that are consistently showing up good across tools that have different approaches and trained on different data sets, that's a really good sign. Let me just look at these questions. Do you have any indications of what type of targets the CNA performs best? No, I mean, so kinases, but that's because there's so much data for them. So it's probably going to be biased towards what there's more data. I do have some the per target data for DUDE, which hopefully you'll be able to see in the paper. But I don't think it can make a really broad answer to that. Have you tried gene family focused CNN scoring, perhaps after aligning proteins? Okay, so the goal of all the scoring in GNINA is to be general purpose. And actually that's a good, what we're about to do now is to make not a general purpose scoring function, right? And certainly other people have, and ourselves have created target specific scoring functions, both CNN scoring functions, and what I'm going to do here is just an example, an empirical scoring function that is target specific, or you can think of pushing towards the realm of a QSAR model. There's some really interesting work on delta delta G prediction from Gianni Di Fabrides, where they can with relatively little data repurpose their model towards the congeneric series. But we're not going to do the CNN. I'm just going to show you like, okay, let's make sort of a target specific scoring function, how you might do this. So here I have all these results that are actually annotated, active decoy. I run it through score only. And for custom scoring, I give it this everything, where if you remember the weights were all 1.0. So the point of doing this is to get this output, which for each molecule, I have the values of all of those different terms. And so these are structural pairwise terms. Well, some of them are functions of the whole ligand, but most of them are pairwise terms. And so this is the magic Python for just sucking all that data from the output, from the standard output. And now I have a pandas array. And so this is just classical machine learning at this point. I have a bunch of features of my examples, and they have a label, active or decoy. And here's a bunch of numerical features. And so at this point, you can just plug it into your machine learning framework. So like machine learning terminology, I have X and Y. I want to fit X to Y. So here's X. I just got rid of the name and the label. And so here's all my features. And my Y is the label. And you can throw this into your favorite machine learning model. So I'll just do a simple logistic regression. And this is actually doing cross-validation predictions. And then if I plot this, this is actually the result of cross-validation on this model. So if we remember back, the general purpose CNN affinity and the consensus was like 0.64 AUC, and its early enrichment went up to about here or here, right? And so now with this trained model, obviously this is much better. So here's my question to you. Do you think this model will be useful for further screening? Right, so I had annotated data, and now maybe I go and search Molport using that exact same Pharmaco4, do the same procedure, minimize it, then rescore them, and then take those terms, those features, and run them through this model to predict. This is a classifier, so it's just saying active or not. Fun to see how many people have dropped off. I'm going to say, yeah, what did you say? Huh, very positive people. So I cannot bring myself to say yes, because anytime I see results this good in a machine learning model, I assume there's something wrong or that it won't generalize. I just can't bring myself to say yes. I think what you would find is it'd be very good at finding hits that look like your training set, that they're very rigid molecules, and are very similar. You would have to evaluate this more rigorously, and like, for example, see, does this actually do anything better than just similarity search to your training set? I'm just skeptical of very good results like this, even though this is cross-validation. But one of the nice things about doing this instead of retraining a CNN model, and I didn't think I'd have time to go into this, and I was right, is you can actually dive into this model. You can interrogate this model. You can see what the weights are in that logistic regression. And I did do that, and it's looking for like rigidity, right? And how useful is that actually is a screening tool. And the other thing, which I can't fully explain, is it's really likes electrostatics and hydrogen bonds, which makes a lot of sense, except that when you look at the sign, it likes molecules that have bad electrostatic interactions and bad hydrogen bond, like acceptor, donor, donor. It, for whatever reason, in this set, it's pulling out things with electrostatic clashes, which, you know, maybe you could think of, we'd have to dive into see what's going on. But it's like, if it's a good molecule, it can tolerate that, and the bad molecules can't. I don't know. But that makes me leery of the molecule, of the model. But I can do that with this model because I have named understandable terms. So I just encourage you to be skeptical. You'd probably want to cross-validate this more rigorously. This is just random. You know, make sure it works on different scaffolds. Like you could look at flexible versus rigid molecules. And of course, all it's learning is the signal needed to separate the decoys from the actives. If in the library actually screening looks very different than the decoys, then it's not a very useful signal. And just as a general rule of thumb, if it's a structure-based model, it should be pose sensitive. If you have an active compound and you put it in the wrong way, it should not still be scored well. The final thing, and this is in bold, is it's tempting to do something like this, to train this sort of model. And like if you train a linear model, you can just create a custom scoring file. You can give it to GNINA and say, go ahead, dock with this custom score. Go ahead, minimize with this custom score. And that's a bad idea because here you're training classification, active versus decoy. It doesn't know about the bad poses, right? So to train a scoring function for pose prediction, you need to actually have as input good and bad poses of the same molecule, right? So I would not take a learned model from this sort of data and use it to change the location of the pose. You won't get good results. So finally, you can provide your own CNN models. I think that's another workshop in its own almost. But we do have scripts and documentation for doing that. As with any machine learning exercise, the hardest part is getting your data into the model. That's not too much of an exaggeration. But the way this is set up is if you can come up with a text file where each row has an example that you're training from that has the label, the affinity, the receptor, and the ligand, you can give that to our scripts and you can train a CNN model, and then you can give it on the command line to GNINA and it can rescore with that model. And I apologize if this is what you're hoping to learn from this workshop. So finally, if you run into trouble, my preferred way for you to see tech support, other than figuring it out on your own, is through our Slack channel. So you can subscribe to this and you can ask questions. If you run into an actual bug or if you have a feature request, the GitHub issues page is the place to go. And this notebook is if you look at the URL, I don't know why it doesn't show the URL anymore. This is hosted on GitHub. The slides are checked into GitHub along with the notebook that generated them. They're in GNINA docs RCS workshop. So thank you. I hope there's still some questions. Yep. Well, let me do the William Pitt. Really? Oh, I mean, he founded University of Pittsburgh. Okay, so transfer learning, that was an example I was talking about with that delta G position or paper. So that might be something a good reason to train a CNN model is to take one of our pre-trained models. And like if you have this ER alpha data set for the transfer learning, what you do is you start with a pre-trained model and you just tweak and refine that model on the ER data. I will say, like, we've tried this, and at least I think it was factor 10A. It didn't actually work very well in our hands for that one example. So if you're doing that, make sure you have a way to evaluate whether it's working or not. Would be my advice. But other people have published various transfer learning with CNN models. When no Pharmaco4 is available for an active site, rather than docking millions of compounds, is there a fragment library that can be done? Okay, that's a great question. So Pharmaco4 elucidation is its own topic. I will tell you what we do, and there's certainly published approaches that are more sophisticated. We use GNINA and we dock fragments. And when I say fragments, I mean really simple fragments like benzene, water, maybe a few other really tiny things. And then our current process is to do this and stare at it and think about it and decide what we think the good interactions are, which is someone who does methodology development. I can't say is a process that makes me happy, but that is what we do. GNINA can, so you can install and run GNINA without a graphics card. You must have the CUDA library installed to build it. If you just, so first of all, it's Linux only. Although you might be able to get, I think some people are working on Windows, but if you just download this mammoth executable, it already has CUDA, so you don't have to worry about it. It just will be slower without the NVIDIA graphics card. Mac is, you can run SMINA on Mac. I mean, NVIDIA is not supported anymore on Mac, so sad. Like we are tied to NVIDIA. But if you just,
I think we're going to have to finish now, actually. We've gone over the time. Been an absolutely fantastic presentation. If people have more questions, I presume they can just post them on the Slack channel.
Yeah.
Well, thank you again, David. This has been, another stunning presentation, and I think it's really fantastic that you've shown us also sort of unpublished results, should I say, for examples. Just shows you that this sort of thing is, whereas docking has been around for a long while, there are still lots of things that need to be sorted out, and so it's great to see these hot off the press news.
Yeah. Also, when your graduate student graduates before finishing the paper, it tends to be unpublished results for longer.
Well, thank you very much anyway. And as I say, I'm sure people have got questions. I'll get in touch with you on the Slack channel.
Great. Thank you for inviting me.
Yeah, it's been brilliant. Thanks very much. And thank you very much to Liverpool Chirochem for sponsoring these events. Okay, I'm going to have to close now, I'm afraid. And hopefully we'll see everybody back next month for the next workshop. Once again, thanks very much. Bye.